Dear friends, dear colleagues, I'm very glad to introduce next speaker from United States, from New York. In New York now, 5 a.m. Professor Dr. Farber, you're welcome.
My presentation is dynamic self-adapting chemical structures and their properties, based on the method of partial combinatorial modification of polyacceptor substances. Today we are scientists and doctors. Tomorrow we may be patients. And I demonstrate this presentation from point of view of adaptability of different systems. I schematically visualize the degree of adaptability of static classical drugs with unchangeable structure. In the human population is a blue circle with a small area proportional to adaptability. The green circle shows the effect of the human organism sleeping due to organism aging and renewal of its cells. It overlaps the blue circle. Due to the slippage effect, cardiologists have to change the combination of hypertensive drugs in people after a while, but their static conservative structure again falls into the blue circle. From another side, microorganisms, fungi, and viruses variability in the gray circle overlap the variability of static drugs and forces pharmaceutical companies to develop new antibiotics. Since the variability of the gray circle cover all circles, we are focused to deal with problems after they arise. For instance, to fight new viruses, it takes many years to create and approve new drugs, but it takes many lives during this time. Since the gray circle is more flexible, the virus mutates again at the same time of new drug creation, etc. It looks like a vicious circle. We don't know what is happening behind the doors of nature's lab. I will use my favorite movies, Gladiator with Russell Crowe, Maximus, to illustrate this point. He said, whatever comes out of this gate, we've got better chance to survival if you are ready to fight. How can this problem be resolved? Actually, our goal is a red circle, which shows the variability and adaptability of our proposed dynamic drugs with self-adaptation capabilities. These circles significantly overlap all possible circles, offering a more adaptable treatment approach than static drugs. Dynamic drugs adapt to particular patient's human body, viruses, bacteria, and cancer cell even faster and more efficiently, offering a promising future in drug development and treatment when the area of the circle will be even more increased. What is the principle of classical drugs with conservative structure? I can represent like a model of one key to one lock. The main idea behind the creation of dynamic drugs is to use a set of lockpicks instead of one key for one lock, which represent a group of many similar molecules that open many locks and adapt to the target. This ensures significantly increased efficiency and the widest possible spectrum of drugs action. Principle of selective specific hybridization of isolated DNA RNA. In this slide, DNA temperature denaturation separate the double helix into single strands into single strands. Modification, both single strands are acylated and reversing mononucleotide charges. Modified chain selectively hybridize only with this precursor. For instance, chain A hybridize only with acylated chain A, and respectively chain B hybridize only with acylated chain B. This process demonstrates how a charge reversal through acylation creates highly specific hybridization patterns, which could be useful in molecular biology application and targeted therapies. This is a principle of creating anti-cancer MolRx. We name this our substance we create, MolRx, from molecular robots. A yeast transplant tRNA, such as alanine tRNA, is taken and treated with nuclease, which leads to the formation of multiple oligonucleotides fragments. The resulting fragments are then modified by acylation. These fragments modified by acylation reverse the charges of oligonucleotide residue by carboxylation. MolRx is a novel anti-cancer drug composed of carboxylated RNA fragments. If such a mixture of carboxylated oligonucleotides, MolRx, is mixed with tRNA, the complementary acylated fragments of the nucleotides from bonds, they form bonds together with their non-acylated precursors and precipitate on the tRNA, disrupting the later functioning and stopping protein synthesis. In this small animation, you can see that cancer cell with active micropinocytosis selectively uptake oligometric RNA fragments with altered charges, MolRx, our substance, from the extracellular space, leading to their demise. Selective uptake, they usually don't normal cells with little or no micropinocytosis remain unaffected. Internalized MolRx fragments interfere with protein synthesis in cancer cells. Disruption of protein synthesis results in cancer cell death. This approach leverages in changes macropinocytosis of cancer cell to deliver MolRx, specifically disrupting vital processes in cancer while minimizing the impact on the normal cells. And in this animation, you can see that without protein synthesis, process of apoptosis in cancer cell triggered by MolRx, impact of protein synthesis. One of examples of our research evaluated several anti-cancer drugs and a bacterium-derived immunomodulatory lectin in treating multi-drug resistant, very serious carcinoma, Lewis carcinoma in mice. MolRx combined with immunomodulatory lectin yield the best outcome with 100% survival in 40 days post-inoculation of the group receiving both. This line, 100% survival. MolRx outperform other tested anti-cancer drugs. For instance, if you look at this cisplatin, its rate about 27%, which we use today. This result highlights MolRx potential as a promising strategy for enhancing cancer therapy, especially powerful approach to improving cancer treatment outcomes, potentially overcoming the limitation of traditional therapies. Here we have properties of MolRx, our dynamic anti-cancer drugs. I just mentioned that It has hundreds of sound-isolated oligoRNAs, like a dynamic structure, and can be adapted to different types of cancer related to adenocarcinomas. The next our project, we use is again our approach of dynamic structure for Albuvir. Albuvir is dynamic drug that blocks the operating mechanism of pathogenic microorganisms. In order to understand how it works, schematically I will show animation. Animation depicts viral genome penetration through the cell membrane. The cell membrane surface contains nuclear pores with receptors for alpha and beta importins. Viruses possesses importin-like proteins, enable them to activate receptors in the cell nucleus. This allows them to open nuclear pores and penetrate into the nucleus. This infection mechanism is characteristic of 80% of all viruses. By inhibiting this mechanism, it may be possible to combat more than 80% of all known viral infections. It potentially leads to broad-spectrum antiviral strategies targeting nuclear entry. Albuvir is represented here by red. React with peptide signal of viral importins, block them, and the virus does not enter the nucleoli where it replicates. Property of Albuvir. The most interesting property of Albuvir is ability to adapt to many viruses that directly or indirectly use the cell nucleus. These are influenza, herpes viruses, coronavirus. Albuvir demonstrates rapid onset, stopping influenza virus replication within 30 minutes when administered orally. 30 minutes and it stopped it. Another project, briefly, oral vaccines mechanism of action. For brevity, when a dynamic vaccine created, somewhere behind of this is already created, by partial modification of antigen, it generates thousands of variants with slightly altered surfaces architectonics. And modified, if I look at this branch, modified antigen, this one, protected from enzymatic hydrolysis, break down into immunogenic haptens. Haptens absorbed into the bloodstream. Absorbed haptens induce an immune response. This approach enables oral vaccine administration while maintaining immunogenicity and overcoming traditional challenges associated with oral vaccine. For comparison, if we use native antigen, hydrolysis by enzyme to amino acids resulting after hydrolysis, they don't induce immune response. That's why we can't use these vaccines orally. When we have, if you apply a few modifiers, not one, few modifiers, we end up with combinatorial mixture of hundreds of thousands of proteins with minor structure and surface charges differently. Many new immunoglobulin monoclines will be produced for such new antigen epitopes, and accordingly, the vaccine will be much more effective. During incomplete partial modification, modifying 2 to 10% in this area of surface positively charged amino acid residues, for instance, for influenza neuraminidase, which has 216 available groups, lead to mixture of, can you imagine this number, 2 to power 216 combination of the molecules that will be modified. At least one compound from this vast majority of components will match the receptor of the vaccine antigen, and the vaccine will be much more effective. Remember our red circle in the beginning of our presentation. No wonder this overlap all the circles, because dynamic adaptability overlapping all the circles based on these huge options. And experiment, for instance, with Pseudomonas antigen revealed that modification of 3%, here, 3%, this line is oscillation, and this antibody titer index of antigen lead to significant increase in the titer of induced antibodies and the appearance of antibodies upon oral administration. It means with the 3% of oscillation, efficacy of this vaccine increasing 4 times. From another side, this oscillated vaccine, if you use this yellow curve, if you use it orally, it works, first of all. And second of all, if you see a maximum for this, matching the results of applying regular vaccine when we have 0 level of oscillation for injection vaccine. So this opens us a way to use many vaccines perorally, and they work and show the same result. But if you modify, it would be even more. Another our project is dynamic insulin. The process of creating dynamic insulin involves several innovative steps that modify the structure and properties of insulin. If insulin is fragmented by highly specific protease, into large fragments, which are then combinatorially modified, a mixture of thousands of oscillated fragments is formed, complementary to the insulin receptors, which is easily absorbed from the intestine into the blood. And in experimental on rats with alaxan-induced diabetes, a single oral administration, of such a mixture of peptides stabilize the glucose level in the rat's blood almost in the normal level within 24 hours. Almost normal, 24 hours. The rapid and significant reduction of blood glucose level within just 1 hour, this 1 hour, of administration is particularly noteworthy. We practice with separate different ingredients, and now we decided, based on experience with individual dynamic drugs, we explore combination multiple dynamic structure to create a second derivative, nanoparticles with potentially enhanced efficacy and synergistic effect. Antiviral nanoparticles composed of multiple dynamic structure. For instance, core based on dynamic cobalamin, these green pentagons. Surface dynamic dipyridamole. Intermediate layer combination oligopeptides, amino acids, and dynamic riboflavin, this intermediate level, embedded between core and structure molecules. This complex structure was predicted to exhibit a broad spectrum of antiviral activity, potentially leveraging the unique property of each component to enhance overall efficacy. Combining different dynamic drugs in a single nanoparticle could lead to synergistic effect, improving antiviral performance beyond what individual components might achieve alone. This approach represents a novel strategy in antiviral drug development, exploiting the versatility of dynamic structure to create more potent and adaptable therapeutic agent. Again, I will pay attention to these ingredients. Very briefly, I just mentioned result of modification of each particular. What we have here again, dipyridamole, riboflavin, and cobalamin. And in this case, when we modify dynamization dipyridamole derivatives using a single modifier, okay, succinic anhydride, 5 new derivatives are formed. When you use 2 modifiers, 12 new derivatives are formed. When we apply this approach using two modifiers significantly increase the number of different derivatives of the original molecules. For instance, riboflavin modified simultaneously with succinic and maleic anhydrides produce 80 derivative reactions with each other. Very important that this diverse modification is achievable only with precise calculation of modified quantities relative, in this case, to riboflavin amount by our method. So very important to have specific way of counting, and we created this method for any substances we have worked so far. When multiple dynamic structures, such as scopolamine, dipyridamole, and riboflavin, are separately synthesized and mixed, the resulting nanoparticles aggregate and successfully capture both by cancer cells and virus-infected cells. They become very powerful. A few words: HPLC chromatograms of initial riboflavin and its initial and its supramolecular structure, and the original dipyridamole and its dynamic structure. This feature demonstrates supramolecular structure formation while preserving essential property. You see, we have many different sets of ingredients compared to what we have here. Similar nature. This significantly departure from traditional static drugs formulation. Our various projects based on dynamic drugs showcase the versatility and potential of the innovative approach to addressing various medical needs. Rather than tailoring drugs before administration, this medication would self-personalize within the patient body. This allows for affordable mass production from one side, while still achieving personalized effect precision in the human body. And it could be applicable to any kind of modern medicine and work by itself. We accomplish, using the dynamic approach, we accomplish a very important problem we solve. Block the development of drug resistance, because static drugs, they're not changeable, they can't do this. In addition, we get set of great properties like wide action spectrum, but low side effect, mass production, and personalization, which is affordable, effective for entire human population and effective for animal. In order to do this, we created the first time in the world, we created a theory of dynamic self-adapting chemical structure and a concept of their creation, including mathematical apparatus to calculate and modeling have been developed. Based on this concept, method for synthesizing dynamic chemical structures through partial modification of polar acceptor substances, name of our method, have been developed. This innovation approach has been demonstrated to be applied to any chemical structure with 3 or more accessible groups that can be modified, for example, by carboxylation of amino groups. This structure uniquely formed with a specific ratio of components calculated according to our proposal mathematical formula with the framework of the theory of dynamic chemical structure. In addition, it has been shown that the proposed structure can be applied in creating dynamic self-organizing and self-adapting drugs. We name it dynamic drugs. Dynamic drugs were synthesized based on dynamic chemical structures. This drug possesses self-organization and self-adaptation properties and can overcome drug resistance developed by the human organism, microorganisms, viruses, and cancer cells. We get a set of the first in the world first set of patents for dynamic structures in different countries. We published articles, and this is some of them in main magazine about this discovery. And the world first dynamic substances In this 30-minute presentation, condensed 30 years of research in dynamic chemical structures and their application in self-adjusted, self-adapted dynamic medical drugs, opening vast possibilities and health treatment. I began the presentation by noting that while we are scientists today, we might be patient tomorrow. Driving our mission to enhance health and longevity, this led to creation of new generation of self-adjusted dynamic medical drugs, resulting into granted patent for longevity, anti-aging, and rejuvenation. So far, we develop and synthesize various dynamic chemical structures, giving us extremely powerful tools for fighting disease and staying healthy. And they relate to antiviral, albuvir, molvrex, nanoparticles, vaccines, anti-cancer, molrex preparat, hemostatics. We use regeneration for stimulus, for that we name it dimazole, antimicrobial, immunomodulators, and neuroprotectors. What future of our envision? Like the Little Prince box containing the perfect sheep, our research offers endless opportunities to those sharing our dreams. Everybody could imagine their dreams of a bright future in this magic box. I encompass my vision of endless innovation in adaptive medical aimed to improving quality of life and longevity. As we age and gain experience, our wisdom becomes mankind's heritage to be passed out to younger generation. For this, we must live longer, healthier, and happier. I would like to acknowledge Professor Artur Martynov, colleagues, and friends with whom I have collaborated for years and whose contributions have been invaluable. This slide represents our optimistic vision. We believe the dynamic self-adapting chemical structure will help millions to be healthier and live longer and happier. Thank you for attention.